A quick note, this session is being recorded, as you know, so that anyone who needs access later can catch up and continue learning. Now, without further ado, let me introduce our incredible moderators who will be guiding today's session. Olasheni Akinriande and Oluwase Yodwole, over to you.
Thank you. Thank you very much, Dr. Lakunle Jaisimi. And thank you once again to MetabHole and DRTC for putting this. My name is Sheni Akinriande, and the tone has been set by Lakunle Jaisimi as well as Alimat Olaniyan. And I guess we are ready to start the program. We should remember that the session is being recorded, and as much as possible, we need to all cooperate so that we can finish on time. And I'll be co-moderating with Sheyi Oduole, and I would like to invite him to introduce himself.
Thank you so much, Sheni. Once again, my name is Oluwase Yodwole. So I'm going to be presenting the first speaker, but before then, I would like to read his professional profile to us so that we can acquaint ourselves with the person that is presenting. Our first speaker today is Dr. Anelize Bauermeister. Please kindly pardon me for the pronunciation if I didn't get that correctly. Dr. Anelize Bauermeister is a professor at the Chemistry Institute of the University of Sao Paulo, Brazil. She graduated with a bachelor's degree in chemistry in 2007 and a PhD in sciences with an emphasis in organic chemistry in the year 2015 at the University of Sao Paulo, USP, and did postdoctoral fellowship at FSF RP/USP Brazil, where she had her first contact with metabolomics at ICB/USP from 2017 to 2019, where she started working with marine organisms. And at the University of California, San Diego from 2019 to 2021 under the supervision of Professor Dr. Peter Dorestein. She has taught workshops and courses on metabolomic tools and is the creator of the Metabolomics Network group on Facebook with more than 700 members from around the world. She is interested in the investigation of metabolites of microbial origin, which are involved in ecological interactions or biological or biotechnological potential. Please, I wanted to make welcome Dr. Anelize Bauermeister for our presentation.
Can you listen to me?
Yes, we can hear you.
Okay, great. Oh, first, thank you, Oluwai, is that correct? Sorry, for the introduction. And also, thanks Olakunle for the invitation. It's an honor to be here. And good afternoon, everyone. Yeah, so good morning for some and good afternoon for others. Today I'll try to talk just a little bit about the application of metabolomics in agriculture and its importance. Next. As definition, metabolomics is the study of all possible metabolites detected in a biological system. And this set of metabolites in a biological system is called the metabolome. Therefore, the application of metabolomics approach can simultaneously perform qualitative and also quantitative analysis of a large number of metabolites. This technique is often used to observe the changes of metabolites in plants under changing environmental conditions and can also be used to study the changes in types and contents of metabolites in different parts of the same plant, comparing different plants, or comparison in different periods. Next. It's very necessary to analyze plant metabolites. The plant endogenous metabolites play an important role in plant growth and development. Sorry. There are more than 200,000 of metabolites in plants, and these metabolites are necessary to maintain plant life and growth, as well as specialized metabolites or secondary metabolites that are closely related to plant disease resistance and stress resistance. To understand and improve crops' stress response and tolerance, researchers have focused on the transcriptional regulation, expression of functional proteins, and the signaling perception, the chemicals, in the stress response mechanism used by plants against abiotic stress. In this scenario, integrated omics analysis centered on metabolomics or integrated metabolomics can be a powerful technique to identify the functions of genes involved in the metabolic process of plants. Only the genomics or the transcriptomics or proteomics analysis, they are all values tools, but you cannot give function for them without the metabolomics. Next, please. Metabolomics has frequently been applied to detect metabolic alterations in plants exposed to various biotic and abiotic stress, including pesticides. Oh, sorry. The widespread use of pesticides and agrochemicals in intensive crop production systems is a serious threat to the functionality and sustainability of agroecosystems. Pesticides accumulation in soil may disrupt soil-plant relationships, thereby posing a pollution risk to agriculture output. So, application of metabolomic techniques in the assessment of the biological consequence of pesticides at the molecular level has emerged as a crucial technique in these investigations. Next. Furthermore, the environment around the plant roots, called rhizosphere, is densely populated with a variety of organisms. Interactions between roots and the rhizosphere community members are mostly achieved via chemical communication. Here are the chemicals again. Chemical compounds. The root exudates contains an array of primary and the specialized metabolites of the plants that can attract, deter, or kill below-ground insect herbivores, nematodes, and microbes, and inhibit competing plants. Therefore, metabolomics of root exudates can potentially help us to better understand this chemical dialogue. Next. Here, I present an example of a metabolomic tool, MicrobeMast. It is a metabolomic tool that we developed to contribute in the investigation of this kind of samples, such as the rhizosphere, to try to understand the chemicals produced by the microbes. So this tool is a database of spectral data produced by microbes, such as bacteria and fungi, and also archaea, that can be used to help in the identification of the microbial metabolites in a dataset such as the rhizosphere, or even in plants. Next, please. So talking about microbes, yes, that's what. So talking about microbes, they are everywhere. And it's estimated that there are around 40 trillion of microbes in our body. And the metabolites produced by these microbes play a key role in several processes and environments. For instance, here, we can see in the left a dysbiosis of two species of bacteria that is three species of bacteria that is already present in our skin. And the dysbiosis against them developed atopic dermatitis. Microbes also are present in the insects world, and in the right we can see that they are also involved in symbiosis relationships by protecting the fungal garden of ants against pathogens by producing compounds with antibiotic activity. Next, please. So these relationships, the symbiosis of microbes with other microbes or even with the host organism is also observed in marine life. And microbes are involved in protection against UV light, pathogens, providing energy, etc. And that's why it's so interesting and important to study and investigate the metabolites involved in this kind of system, just as an example of the rhizosphere. Please, next. So Olakule asked me to talk about the application of metabolomics in agriculture, and I work mainly with the microbes in this microbiome system using metabolomics to investigate the metabolites. So nowadays I'm working in a project applying metabolomics to study the chemicals produced by microbes in marine corals to help understanding the coral bleaching, which has achieved the worldwide in the last few years. And if there are anyone here interested in contributing this project, please let me know and I'll be happy to chat. Next, please. So here's the Rede Metabolomica. Is a group on Facebook that I and two friends created in 2020 for people that are interested in metabolomics content, discussion about tools, to help with doubts, and many other things. So please join if you want. Next. Yeah, so that's what I have to discuss with you today. Metabolomics is a very important tool that can detect and study the set of metabolites in an environment. They could be involved in, they could be present in plant, in the rhizosphere, produced by microbes or produced by the plant, or present in food. It's also very important to check the quality of food. And so it's an amazing tool that has been applied more and more often in the last few years. And there are a lot of tools being developed to analyze this data. So I hope you understand a little bit about application, the possible application of metabolomics here. And thank you all for the interest in this talk and the attention. Thank you again, Olakule, for the invitation and opportunity here. It is a pleasure for me to see you again after a while, right?
Yeah, thank you. Thank you very much. Thank you for your presentation and for accepting to give this talk. Yeah, thank you. We really appreciate it.
Thank you, Dr.
All right.
Please, I want us to actively engage with by using the login. Appreciate Dr. That wonderful presentation. Thank you so much, Dr.
You're welcome.
Thank you, Dr. Anelize, for that presentation on real-world examples and potential research collaborations. metabolomics and applications. Thank you very much. So I guess we have actually started learning bit by bit on the importance of analysis of metabolites, on considering that the plant kingdom has over 200,000 metabolites, and there's a lot to be done with that number of metabolites. The application of metabolomic techniques, as well as the role of microbes in the various ecosystems. Thank you very much once again, Dr. Anelize.